Hey guys, so today we are going to be talking about NovoSeven, which is a drug used to treat hemophilia. Basically, this is just the brand name for it. What it actually is recombinant factor 7A, and this is basically a factor that is heavily involved in blood coagulation and the coagulation cascade. So, as we get started, it's important to talk about what is hemophilia. And basically, this is a genetic disorder that's found on the X chromosome. And what this gene codes for when you have hemophilia is the inhibition of factors 8 and or 9. And these are very important with coagulation and blood clotting. And without them, it can lead to excessive bleeding in dangerous amounts, even fatal amounts. And overall, hemophilia affects one in about 5,000 males, and this is less common in females. And you can see this to the right, basically because it's only found in the X chromosome, because men have one X, one Y. All you need is one affected chromosome to have hemophilia, versus in females, you have two X chromosomes. So if only one is affected, that makes you a carrier. You're able to pass it on to your um, offspring, but only when you have two X chromosomes with that affected gene, then will you actually show and express hemophilia. So this diagram shows kind of the mechanism of hemophilia and just kind of what happens in general with the healthy body versus hemophilic. So top you can see normally if there's an injured blood vessel, what's going to happen is these factors will activate and then lead to platelet plug formation, and then it will clot and stop bleeding. Versus with hemophilia, the clotting factors will not fully activate, and what happens is it won't completely clot and the bleeding will not stop. And looking at our factor, we talked about factors 8 and 9, and the one that we're focusing in on with this drug is factor 7A. And basically this is a vitamin K dependent plasma glycoprotein, and it still is important in coagulation, but instead of direct impact on clotting, it's actually a factor used to activate 8 and 9, which will then lead to the clotting. And this is seen in this diagram. So you have tissue factor that's exposed at some injury site, and then that will bind to factor 7A. And from there on out, that's going to be activated and go on to further activate factors 8 and 9, which will continue the coagulation cascade and lead to the clotting of a wound. So now looking a little bit at the history of treatment for hemophilia up to this point, it all started in 2nd century AD, and that's when it kind of first became known or recognized to some extent. They still didn't really know what it was, but there's some degree of knowledge that something's wrong here. There's excessive bleeding. That's a problem. But it wasn't until the 1800s when a man named John Conrad Otto, he published an article talking about how this unknown disease affected mostly men, and it also was passed on through families. So he kind of started the idea of, one, this is an inherited trait, and two, it's mostly affecting men. And as the years went on, these studies continued, and eventually it was early 1900s when treatments began. At this point, they began to use fresh blood transfusions to treat this and to replenish the blood that was lost through hemophilia. Later on, it developed into exploring treatments such as using bone marrow, thyroid gland, inhaled oxygen, and even diluted snake venom in the 1930s. And in 1937, there were a couple of Harvard physicians that wrote this paper about the presence of globulins in plasma that were important with clotting, and that became an important treatment. Frozen plasma was used for transfusions and treatments for hemophilic patients. They soon realized that this plasma also contained factors 8 and 9, and this was an important part of treatment as those are a crucial part of clotting. So this became a really popular treatment, especially starting in the 60s. Except once they reached the 80s, then they started to realize that people with severe hemophilia were starting to get HIV or hepatitis C because the clotting factors were contaminated. This led to the deaths of thousands, and it made researchers think about how can we change this? What do we have to do to make a new treatment that's not going to cause us death, that's not going to have this risk for us? And at that point, they started looking into new treatments such as NovoSeven. So looking at kind of a big timeline, before NovoSeven, we talked about these treatments they had, and it was in the 1980s that, along with that issue with contamination and deaths, there's also the rise of protein producing technology. And with those two ideas, these two companies, Novo Nordisk and Zymogenetics, they came together to use that technology to produce this recombinant factor 7A as the goal, with the goal of creating this new treatment that won't have that risk of transmitting HIV. So then, March 1988, they successfully treated their first patient with NovoSeven, and it was finally approved by the FDA in 1992. So now we gotta ask, how did they actually make this? How did they go about producing factor 7A? What they used is baby hamster kidney cells. They would take this gene for the human factor 7, and they cloned it as a single chain, and then after that, they secrete it into this culture medium. And once it's in there, they're able to use what's called ion exchange chromatography, which acts as a purification process, but then also creates a two-chain form, which is the active form, and that's what we know as factor 7A. So there's one other player in this, which is hepsin. It's a transmembrane protease, which also has a role in activating certain factors with blood coagulation. We'll talk about it a little bit more, but just want to introduce that. So looking forward at the trial phase and effectiveness of this, they started with some home trials where patients at home, when they had episodes of excessive bleeding or abrasions or anything that happened, they used this drug. And there was a 92% effectiveness rate in clotting and like stopping that bleeding. And this was similar to seen in surgeries where there's a 90% effectiveness rate. This doesn't tell us as much as we would like, and there's been more research done more recently that I will talk about in a few minutes. First, we got to talk about clinical applications. What is this actually used for? One of the biggest reasons this is used is for surgeries and invasive care, and then consistent bleeding episodes, especially in surgery to prevent excessive bleeding and to retain that blood so they don't have to continually transplant blood. As you can see in this chart on the right, it's showing that factor 7, NovoSeven, helps to decrease the amount of blood that's necessary to be transfused during research in a few different manners. So this is really practical and important way that this can be used, along with just consistent bleeding episodes, allowing people to use this, increase clotting, and prevent excessive bleeding. So as I mentioned a minute ago, there are further studies done, and these are better because they are randomized, and there's placebo control, and they're blinded. And they categorize these in two main areas: one, bleeding in surgery, and then two, non-surgical bleeding. So first, looking at surgery for trauma injuries, it was seen that with blunt trauma, this NovoSeven caused a significant reduction in blood transfusion. So that's beneficial because the less blood that needs to be transfused means that it's doing its job of clotting. Versus with penetrating trauma, although there was a reduction in blood transfusion necessary, it wasn't a significant difference from the placebo. Looking at non-trauma related, for prostatectomy, once again, there's a significant reduction in blood loss, which shows as clotting and helping, versus in cardiac surgery, there wasn't significant change. And although there was minimal research on this, they still have to do more research. It's just important to note that although there's reduction, not significant change. Finally, look at non-surgical bleeding, especially with cases of cirrhosis and GI bleeding. This did have positive effects of increasing homeostasis and decreasing failures of these surgeries. And so overall, basically, you can see with all these, there are positive benefits. Each of the surgeries showed a reduction in blood loss, a reduction in the amount of blood needed to be transfused, but whether or not it was significant varied for each surgery or case, if non-surgical. So basically, what do we take away from this? Overall, it seems to be effective, but there are cases that need more research and need to be looked into more to see how effective is this really, and how practical is it? So looking at some new technology that's being developed, it's a fairly similar process, but slightly different ways that they're creating this recombinant factor 7A. And basically what they do is they start with this human hematoma cell line, and from that they extract RNA and run reverse transcription on it, where basically they isolate and amplify the genes associated with factor 7 and also with hepsin, which we talked about earlier, which is involved with clotting and activating those factors 8 and 9. Basically, once they isolate and amplify that complementary DNA, they can prepare it for the mammalian vector, which is basically saying they're preparing it to insert into these Chinese hamster ovary cells. And once that complementary DNA is inserted, it will bind with the Chinese hamster ovary cells DNA, and it will develop and differentiate. And eventually, these recombinant cells can be selected and used for treatment. So now that we're kind of up to speed, I just want to take a second to look at this actual company that started NovoSeven and where they're at now. At this point, they've expanded to obesity and diabetes care, and not just hemophilia care, although it is one of their flagships. Also with hemophilia, now that it's been several decades since factor 7A was first developed, now there's more competitors and it's a better market. And even just for NovoSeven, this market was at 1.36 billion in 2016, which is showing how much it's grown. And as you can see in this diagram, that number is expected to continue to rise. Where we're at right now at 2022, projected to 2027, continuing to grow. So as you can see, it is a big market, and it doesn't seem to be slowing down. And also, just one of the main competitors for NovoSeven is created from Hemobiologics, and they created a product called 7Fact, which is basically the same thing, but just one of those primary competitors. One other thought is the idea of any ethical concerns with this research and development of the recombinant drug. And although there might be some concerns about animal testing, there's no really harm done to these hamsters by taking these cells. And even if there were, the question would be how many lives could be saved for humans at the sacrifice of this. And that's where people fall on different ends, and it's something to be talked about, but overall it's a relatively safe procedure and simple, and overall seems ethically and morally okay. And yeah, thank you once again for listening to my presentation. I hope you liked it, and let me know if you ever have any questions. Thank you.